Okay, guys, this is going to be part one of chapter 12. We're going to start talking about the nervous system, nervous tissue. All right, so as far as the functions associated with the nervous system, um, these are basically what the nerves are going to do. They're going to take input from the senses. They're going to interpret that input, make a decision, and then they're going to output the decision. So, for example, um, if I'm sitting down and it gets uncomfortably hot, the sensors are going to send that information to the brain. It's going to interpret it and then make a decision for me to stand up. And the motor output goes to the skeletal muscles. So whatever sensory receptors we have, internal and external, these are interpreted, and then the appropriate effector organ, either muscles or glands, are activated. So here's just another illustration of it. It's pretty straightforward. You see the water, uh, you decide you're thirsty, you send the you make that decision, the integration, and this is where it happens at the brain. And then you send the output to the skeletal muscles, pick up the glass and bring it to your mouth. Okay, as far as the divisions of the nervous system, two big divisions to start with. One is the central nervous system. The central nervous system consists of the brain and the spinal cord. This is where the integration and the decisions are made. So, brain and spinal cord. The peripheral nerves, basically, it's all the nerves outside the CNS. Specifically, coming from the spine, you have nerves that leave called spinal nerves. In the brain, or I should say in the brain stem, you have these same type of nerves, only we call these cranial nerves. So the spinal nerves come off from the spine below the foramen magnum, and above it we call them cranial nerves. But they do have this in common. They're peripheral nerves. Now, as far as the peripheral division, it too is divided into two major parts. One is our sensory, and it's also called afferent, and we have a motor or an efferent division. So the motor division is pretty easy. It sends or relays the decision made by the central nervous system to whatever effector organ is being called upon. So, for example, again, if we were wanting to pick up that glass of water, I would probably need for my biceps brachii and the brachialis muscle to be activated, and these guys would be the ones who would do so. So that's what the motor or the efferent division does. It sends the go signal to the muscles. And if you recall, that go signal is actually going to cause the release of calcium from the sarcoplasmic reticulum. Now, the sensory or afferent division, we have two branches of these. We've got one that's somatic and one that's visceral. Somatic is going to be giving us information on the skin, skeletal muscles, and joints. The visceral is actually going to give information from the internal organs. This one gets it's when you actually have input from these guys, it can get kind of screwy. I'll explain more about that later. Okay, make sure, yeah, that's everything on there. Okay, the motor division of PNS, again, we've got two divisions. We've got the somatic and we've got the autonomic. Somatic These are the guys that control your skeletal muscles. These are voluntary. The autonomic, or is the involuntary nervous system. Think of autonomic as being almost like automatic. It's not something you consciously control. These are going to be the visceral motor nerve fibers that are going along with those visceral sensory fibers. These guys are going to be in charge of smooth muscle. They are going to have some say in cardiac muscle. Cardiac muscle beats without any sensory innervation, but these guys are either going to speed it up or slow it down. And of course the endocrine glands and the other glands. So these in itself are divided into two subdivisions, sympathetic and parasympathetic nervous systems. So here's a good schematic on this, guys. CNS, you see all this here. You see the somatic sensory fibers from the skin, the visceral. You see the sympathetic motor going to like the heart, and you see the parasympathetic going to the bladder. And I'll tell you a little bit more about these. And then the motor somatic going to the skeletal muscles. So there's the PNS, cranial spinal nerves. We talked about the sensory, the afferent, talked about those guys. We talked about the motor, somatic nervous system, which basically acts on the skeletal muscles. The ANS, or the autonomic nervous system, cardiac, smooth muscle glands. Now, the sympathetic division is the guy that is the fight or flight division. This is the guy that prepares you to either run or fight when you feel like you're in danger. That's what the sympathetic division does. The parasympathetic is also often called the rest and digest division because it is more about processing your food in your GI tract than it is about preparing you for a life or death struggle. Nervous tissue, we're going to be talking more about these in a later chapter, guys. I'm not blowing you off. I'm just don't want to get, bogged down there when we have so much to do. Nervous tissue, this is the communications. Receptors, we've talked about receptors before, and how strong the stimulus is, say if it's temperature or if it's pressure, depends on how the plasma membrane voltage changes. And of course receptors just depend on what they actually measure. So this person is apparently getting ready to get into the shower, is checking the temperature. The sensory ending in the skin is going. It's actually going to come back here to the back part of the spine. From there, it's going to go up. It's going to have a synapse with an interneuron. This interneuron is going to go and it's going to have a synapse here. And this one is finally, this interneuron is going up here to the actual part of the brain that's responsible for interpreting that particular area. So the different parts of our body have different parts of the brain. But basically what we're saying, that's the sensory pathway. Once this is decided, then we have this upper motor neuron. He's going to be the one that sends the command to maybe a little bit more hot, a little bit more cold. It's perfect, pull your hand back, whatever the case. So the upper motor neuron is going, and it's going to synapse in the spinal cord, the anterior part, and its lower motor neuron is going to actually go and innervate those muscles in the hand. Okay, talk about the histology of our nervous tissue. Nervous tissue consists of the neurons. These are the guys that do the communication. But they also have support cells, glial cells, astrocytes, microglia, ependymal cells, oligodendrocytes. Those are all in the CNS. And in the PNS, we have the satellite cells and the Schwann cells. Okay, we're going to take these kind of one at a time. Astrocytes, these are the most numerous of the glia cells, very versatile. These guys have branches that form these processes, and they have these, they call them end feet. These guys are actually going to hold on to the neurons and support them, and they're also going to put their processes around the capillaries, and that's going to help them form what's known as the blood-brain barrier, make sure that only certain things are allowed to leave the blood and enter into the brain. During development, these astrocytes are going to guide the newly formed neurons to their places. Going to have a lot to do with the chemical environment. A lot of that's going to be through their blood-brain barrier, and have some participation in information processes. And just so you know, guys, neurons don't divide. At least no one is, as far as I know, at this current time. So most of the brain cancers you hear about are going to be these glia cells. Astrocytoma is a fairly common brain cancer. Microglia, these guys have the processes on them. These guys are basically the immune system for the brain. These are the immune cells of the brain. They're going to be a phagocyte as far as microorganisms or any kind of debris that needs to be cleared away. They're going to take care of it. So the brain has its own separate immune system. That doesn't mean the other parts of the main immune system can't get there, but if they do, that means something is pretty wrong. Ependymal cells, we'll hear more about these guys. These guys are going to be the guys that help us to form cerebrospinal fluid, CSF. This is what surrounds and bathes the brain and the spinal cord. So these guys take fluid from the capillaries and they filter them and put it there, or allow them to pass through there. In addition, if you see, these guys have got cilia. Those cilia are going to wave and actually cause that CSF to circulate. It's going to go all the way down to the bottom of the spine, all the way back up, and then eventually it's going to rejoin the venous system and the top of the brain. We'll go into more detail about that later as well. The oligodendrocytes, these guys have got the branches, and these processes are going to wrap around part of the neuron. This part of the neuron is called the axon. This is how the nerve cell talks. This is how the nerve cell hears. The dendrites, this is where the axons from other neurons come in and convey messages. But this guy's going to send it out this way. These guys are going to wrap around good long sections, going to leave some gaps, but it's going to wrap around. This is an insulation, kind of like the wire in your house. You have the plastic or rubber insulation around the actual wires themselves to insulate them. We'll talk more about the physiology behind this later, but for right now, it's enough to know that's the insulation. Satellite cells and the Schwann cells. Now this is the peripheral nervous system. The satellite cells are going to surround this nerve cell here, going to protect it to a degree. And the Schwann cells, now let me go back. Do you notice how this one single oligodendrocyte is forming parts of this insulation? By the way, we might as well give this thing a name. Let's call it the myelin sheath. But this single cell is going to form parts of the myelin sheath on maybe several different axons. Now let's contrast this with the myelin sheath here. Here, a single cell is going to wrap himself up like a jelly roll around the axon. So you have a cell there and a cell there. These are separate cells. These are going to form the myelin sheath in the peripheral nervous system. These guys also have a lot to do with regrowing the damaged nerves. So if your nerves get cut in the PNS, there's a possibility that they can regenerate, especially the axons. It's not guaranteed 100%, but the Schwann cells play a big role in that. Again, we'll get into that later. Now we're finally back to the neuron itself. These guys can be 100 years old. You get these guys when you're born and they don't divide. You don't get any more. If you kill them off with drugs or alcohol or whatever, they're gone forever. These guys are amitotic. Some exceptions. Nothing that we need to get in there. Now this is something, these guys have got a high metabolic rate. These guys have got to have continuous supply of blood sugar and oxygen. That's why doing without oxygen for an adult for more than 10 minutes usually results in brain damage, because these neurons will die without that oxygen. They have to have blood sugar, and a good chunk of our body's energy production goes to power the brain. But then the brain controls everything, so it's kind of a super important. You can see in there the mitochondria, the endoplasmic Golgi, same sort of things you would expect. Now there's the cell body. They're coming off, you've got one axon. That axon kind of, as it comes off the cell body, it's got this funnel shape. That has a specific name. It's called the axon hillock. And then here's the axon, which remember is outgoing messages. It's like having one cell phone to receive calls and one cell phone to make calls. And so the axon is our outgoing. Now, of course, when it gets down there, we know from our discussion on skeletal muscles, these guys can split into many synaptic terminals. And here's another piece of terminology. Telodendria are those where the axon splits, and then the axon terminals are, of course, those boutons, the little button-like objects. These guys, of course, are the dendrites. These guys branch. Now, another neuron can form a synapse with these dendrites. You can actually put it right there on the axon hillock or on the cell body. We'll talk more about that later too when we get to the specifics. Here's again a close-up. This guy is also called a perikaryon or soma. It's a cell body. Cytoplasm, microtubules, lysosomes, the normal complement of cell organelles. This is where all the biosynthesis takes place. Got the normal round nucleus, got the Golgi. Now, here's something I have no idea why, but in the neuron, they call the rough ER the Nissl bodies. I don't know why they had to give it a special name. They just did, and we're kind of stuck with it. There's also a network of neurofibrils, neurofilaments that are inside the soma. The axon hillock we've already discussed. Now, when you get a bunch of cell bodies of neurons in the CNS, we call these nuclei. I know it sounds like nucleus. Can be helped. Somebody ran out of creativity when they came to naming these guys. So a lot of cluster of cell bodies in the CNS is a nuclei. In the PNS, we call it a ganglia. Okay, guys, the neuronal processes, the dendrite, short, branch, receptive, and if we have a synapse with the synaptic terminal of a different neuron, then the neurotransmitter floats across and we get the same thing. We'll talk about the gradient potentials as well. So the axon, this guy, the sends an electrical signal down, and it's definitely a one-way street. There's the myelin sheath, and these cleared areas where there are no myelin sheath are called the nodes of Ranvier. One axon per cell. Some of these guys can be very long. There is axons that are 3 feet long, because the nerve bodies that for the nerves in your feet are at the bottom of the spine, which is above the hip. So from there, way up there, they send this axon all the way down to your feet, about 3 feet in distance for most folks, I guess. Anyway, the axon collaterals, some of those come off, and then the teledendria we've already talked about. These axon terminals, I was taught Bhutan, so if you hear me say Bhutan, that's just me. You can call them any one of these 3 names. Secretory region, in other words, this is the place from which the neurotransmitter is released. Remember, we talked about that when we were talking about the neuromuscular junction. So these guys can release neurotransmitter to talk to another neuron, or if this is a motor neuron, it's going to be to a muscle or a gland. Now, we'll talk more about this as well, but when we release those neurotransmitters, you can get some neurotransmitters that are excitatory, and you get some that are actually inhibitory that kind of make it more difficult for something to happen. And we'll talk about that when we get further on. Functions of the axon is the conducting region. That's where the electrical input pulse leaves the cell body, goes all the way to there to the axon terminal. Now, the electrical signal is one way. However, since we said that this cell body is where all the biosynthesis takes place, it doesn't make sense to you guys that if something wears out in the axon, it's going to have to be transported back up here to be recycled, and something new is going to have to be transported down this way to replace it. That's what anterograde transport means, moving from the body to down the axon. Retrograde transport is what I was telling you. If you have something that's worn out that needs to be recycled, you send it. By the way, that is the way that this little virus right here gets from your periphery into your brain. And his name is rabies. The rabies virus actually uses the retrograde transport in your axon to get there and eventually make its way to the brain and all the nasty stuff that happens. So anyway, retrograde, anterograde, you got it. The myelin sheath, protein and lipoid. It's got a lot of lipids in it. Not every axon is going to have it. They're all not going to be insulated to the same degree. Some of them are insulated more than others. We'll talk about that when we get down there. For right now, let's just talk about it. This is how we're going to send the electrical signal. So on the areas where you have this glial cell, whether it's a Schwann cell or a oligodendrocyte, doesn't really matter. There is no way for sodium to come in, and we know that when we're conducting an impulse, we're basically allowing sodium to come in and changing this guy from negative to positive. So what happens is only at these nodes of Ranvier can this sodium come in. It comes in and it changes, and then it's not going to be able to open new gates until it gets to the next node. So that's why, let me see if I can illustrate this for you guys. That's why these guys, the signal, it doesn't really jump, but it has the effect of jumping because there's only certain places where the sodium gates can be opened. This guy is going to have like a jump. Why would you want to do that? Do you know that down in Australia, they have kangaroos, and these guys can jump 10 feet with one jump? Do you think that kangaroo can jump 10 feet faster than I can run 10 feet? The answer is yes. It's faster. It speeds up conduction. That's why it's there. We'll talk more about this. Is called saltatory conduction. We'll get to that again coming up. Myelin sheath in the CNS we've already talked about. The oligodendrocytes has processes. The whole cell doesn't wrap around. We do have nodes of Ranvier, but some of the thinnest of the axon fibers don't have myelination, and some of them don't. It depends on how fast we need the nerves to be. Not all nerves need to be fast. Some of them can be relatively slower and still work just fine. Okay, the myelin sheath in the PNS, the Schwann cells, and so the Schwann cell starts wrapping around, and he wraps around and around, and as he wraps and he pulls tighter, then he kind of pushes a large part of the cytoplasm and the nucleus up there. So it's really like one of those jelly rolls that you see like in a bakery, just like that. And so there's one cell, there's a second one, and a third one, and so forth. And then the gaps between these are going to be our nodes of Ranvier. I have no idea what this is doing there, guys. Anyway, nodes of Ranvier, the gaps, and if we have any axon collaterals, they'll be coming out there. We talked about the thin fibers. One Schwann cell may incompletely enclose 15 or more unmyelinated axons. It doesn't make much sense either. The peripheral bulge of the cytoplasm we've already referred to. I have no idea what that is. All right, guys, that's the end of part one. I'll come back with part two very shortly.